Many researchers in cancer and many other fields of biomedical research are using cell cultures. So they're growing cells in flasks in the laboratory as a way of manipulating cells, looking at responses to cells, and understanding how cells and genes and their products work. Now in these cultures, cell death also occurs. And it can occur by suicidal modes that one sees in the body, but also through accidental modes, through shear forces that these cells might suffer. Freezing cells for storage and thawing them, that causes cell death. So there are many reasons for cell death in culture. But the problem with cultures like this is that they are lacking the phagocytes that do the clearance, that critical job that keeps our tissues in the body healthy. So I got to wondering some years ago about whether it would be possible to design a means by which one could substitute for the phagocytes and selectively pull out in an efficient way non-viable cells, the dying and the dead cells from these cultures which lack phagocytes. And so, cutting a long story short, we came up with an idea that was to design a particle which is in suspension in this little tube here, which is a super paramagnetic particle, which means that in a magnetic field becomes highly magnetized. And these particles we've developed to have a surface which can discriminate between viable and non-viable cells but by a short period of incubation with the cells. So one can take these particles, incubate them with cells, and essentially place them in a small tube together, and subsequently separate the particles with the non-viable cells attached on a very simple but powerful magnet. And one can take away the viable cells that are not separated in the magnetic field. And in this way, one can purify viable populations of cells very simply and very efficiently. And essentially, the viable cells are not touched by the procedure. So it's a negative selection procedure that's very useful for many applications in biomedical research science. What we do is we take an aliquot of particles, add to the cells, and incubate typically for around 30 minutes. During the incubation period, the particles have the opportunity to come into contact with cells in the mixture. If they come into contact with non-viable cells, they will bind very avidly to those non-viable cells. If they come into contact with viable cells, they will not bind. And so during the incubation period, the non-viable cells collect particles. Now after the incubation period, one essentially adds some medium, and typically we would be adding a volume of medium that makes the total volume up to 1 ml, and resuspend the mixture of particles and cells so that you can place them in the magnetic field, and this is a very simple but powerful neodymium magnet, and then very quickly, over a period of 3 minutes, allow that separation to occur, in which the particles are drawn to the side of the tube next to the magnet. So the viable cells are left behind, the particles that are not bound to anything get drawn to the magnet, and the particles that are bound to the non-viable cells drag the non-viable cells to the wall of the tube next to the magnet, and with a simple pipetting action one can just remove the suspension containing the vibrant viable cells. Anyone who's using cells is dealing with cell death and needs to deal with cell death, and often that cell death is ignored because it's difficult to deal with it, or people sometimes don't even realize the level of cell death that's in their cultures. A very common practice is to produce antibodies in culture, and they can be used for many purposes. They could be used for research, they can be used for diagnostics, they can be used for therapeutics. we've found that using this technology, you can improve your yield of antibody by 100%. You can rapidly kickstart cells that have been stored frozen so that they establish much more rapidly in culture, thus saving you time for your research. It gives you more cells in a shorter period of time. You can establish populations of cells for cell-based drug screening assays, for instance, that are much more reproducible because the viability is always high. You can improve the products of cells. I've mentioned antibodies being produced from cell cultures. You can produce more cells. Stem cells, for instance, very popular type of cell. And if you can get more stem cells more quickly for your research, then you need to do it, and now you can do it. If you've got cells of the highest viability to start with, which you can do with this simple technology, then it means that your variation between experiments is reduced as well, and that all helps to accelerate your research.